Good morning, good afternoon, or good evening, depending on where you are in the world, and welcome to today's MDDI QMed webinar, A Year of MDR Remediation, Strategies and Lessons Learned, sponsored by Nelson Labs and broadcast by Informa. I'm Daphne Allen, and I'll be your moderator today. We have just a few announcements before we begin. This webinar is designed to be interactive. The dock of widgets at the bottom of your screen will allow you to learn about today's speakers, download resources, share this webinar via social media outlets, and participate in the Q&A session that takes place at the end of our presentation. The slides will advance automatically throughout the event. You may also download a copy of the slides via the Resources widget. Toward the end of our webinar, we will ask you to complete our survey found on the right-hand side of your screen. Please take a minute to fill this out before leaving us today, as your feedback will provide us with valuable information on how we can improve future events. Lastly, if you're experiencing any technical problems, please click the Help widget found at the bottom of your screen, or type your issue into the Q&A area, and we will be glad to offer one-on-one assistance. Now, onto the presentation, A Year of MDR Remediation, Strategies and Lessons Learned. Discussing today's topic are Jennifer Braunschweig, MS, Engineering Director for Medtronic, and Matthew Jorgensen, PhD, DABT, Senior Chemist and Toxicologist for Nelson Labs. Now, over to you, Matt.
Thanks, Daphne, for that introduction. I really appreciate it, and it's really great to be here to do another one of these webinars. I always love the opportunity to hop on the line and share some of our experiences, so that that all of us together as a medical device community can improve and know how to navigate these tricky times better. So I'm absolutely happy to be here. It's been a super exciting and kind of a crazy time in the world of medical device with all of the changes that have been happening. Of course, we know that the background here with the new MDR, we've been talking about this now for a couple of years, leading up to the May 2020 deadline that was recently postponed. Of course, along with that, we have new standards that we're trying to understand and we're kind of like adapting to. Of course, that's the new 10993-18, the new 10993-1, and then even though it's not that recent right now, the ISO 18562 is still one of these that we're kind of coming to grips with and interpreting from a regulatory perspective, that how it applies to different types of gas path devices and so on and so forth. But with this regular kind of crazy background of all of these regulations changing and new standards in place and new notified bodies becoming certified under the new MDR and so on and so forth, then we get this curveball, SARS-CoV-2 spreading across the globe that basically turned everything upside down. It's the cause of the MDR deadline being delayed, and at the same time has been has caused everybody to rapidly shift some of their priorities during this time. So I can say from the Nelson Labs side, certainly we were really fully focused, especially in our advisory services group, on helping people understand and prepare for the new MDR, and then with the COVID-19 pandemic, pivoted quickly to focusing on helping in whatever way we can to aid companies as they develop devices and PPE and all of this stuff to aid in the pandemic. So it's been a really dynamic time. A lot of interesting things going on. With the deadline postponed for the MDR, it is kind of a good time to take a step back and think about some of the lessons that we've learned so far, what I wanted to do with these slides is kind of go through some of the process that we have for an MDR remediation, or when we help folks with their MDR remediations, and along the way kind of expand on some of the things that we've learned in the last year. So, of course, this is the timeline that we've all seen before. May 2020 was the deadline for new device submissions under the new MDR. That was pushed back a year. I think that it's worthwhile just to remind everybody that while this first deadline was pushed back a year, that subsequent deadlines weren't pushed back a year. So that means that there's still going to be a lot of tightness there down the road as we approach May 2024 and May 2025, as the deadlines where all devices on the market must be MDR compliant. Of course, with the new MDR, the pinch points that we've been dealing with are this no grandfathering provision. That's been something that has been a difference that's caused maybe some of the most heartburn as companies go through their remediation process. There's an expanded definition of what is considered a medical device, and then there's also this requirement of a qualified person who is responsible for compliance. So really, this MDR remediation process, it's all about biocompatibility, or at least that's the way that we see it. Of course, there's other elements, but the biggest push and the amount of work has been related to going over all of the documentation and files related to devices on the market, and doing like a biocompatibility gap analysis where we gain an understanding of where those gaps in what was done in the past versus the state-of-the-art standard are, to determine whether or not there's any testing or further evaluations need to be done. So that's the main thing there. It's with biocompatibility. Of course, as we go through that, we're comparing this or looking at what was done in the past in the context of the most state-of-the-art standards. So that would be the new ISO 10993-1. So you can see a screenshot of this here. So this was issued in August 2018, and you can see that it changed a few things that are pretty impactful to this process. One being that the biological endpoints that are needed to be addressed are all marked with E's for evaluate rather than X's, which sort of led people down this path to think that testing was really required to address all of those. There's a new column in there, the physical and/or chemical characterization information, and there's an X there, and that in contrast to the E, that really is made to indicate that sponsors should know, that we all should know really what are our medical devices made out of, what type of chemicals might they leach. It doesn't necessarily, or wasn't intended to mean or indicate that chemistry testing is required for all devices. So I think that we found in our conversations with notified bodies that a lot of those notified bodies are really kind of interpreting those X's to mean that some testing should be done, at least when it comes to chemistry and material information. So this is what this is the new context for these evaluations. This and the new ISO 10993-18, which was just finalized this year. As we go through that, we really use a risk-based approach within the context of 10993, looking at all of the documentation available to do a gap analysis, and then make what we call a biological evaluation plan. So I'm calling this a biological evaluation plan here. Really, you could also call this like an MDR gap analysis. But really the main function of this process is to, after all of the information on an existing device is collected, to review that information and try to understand and prescribe what type of testing really needs to be done, testing or evaluations, additional evaluations need to be done in order to bring that device into MDR compliance. And this is one of these things where we've really learned a lot over the last year. So I could say if we were to have this same conversation 1 year ago, the experience that I would relate is that we had been talking to quite a few companies about their MDR remediation plans, and most companies were coming in kind of like guns a-blazing, like really ready to do quite a lot of testing. So they had budgeted and prescribed and planned on doing really a huge volume of testing in order to bring their devices into MDR compliance. However, once these companies have really gotten into this process and dug deeper, and with our help too, right? So Nielsen Labs as an advisory service can come in and help with this process. What we've really found is that actually a lot less testing has been necessary in order to bring devices into compliance than we expected a year ago. So I would say that as while initially things can look kind of bleak and like it's going to be very, very expensive and extremely long and painful, while it still is, painful, it might be that there's a lot less biological testing that's really required at all. So I would say that that's definitely one clear lesson learned, is that the testing plans have been scaled back a lot as it's been realized that actually a lot of the documentation and files that we have for devices on the market in Europe aren't that bad, and that by remediating these files and doing a proper gap assessment, the gaps are actually pretty small. One of the other things that we look at in this document, so first of all, we go through, we review everything that's been there or that's existing regarding the biocompatibility of a device, and then we'll go through and start to prescribe a testing plan and a family grouping strategy, and I want to talk about that a little bit too here. So depending on the nature of the device contact type, different biological endpoints are required for evaluation. So that's clear from ISO 10993-1, and there's different options available to evaluate or address those biological endpoints that are required, right? And so that's one of the things that I want to make sure is totally clear, is that in any of these cases there are options, and we see companies, depending on their level of risk aversion and regulatory risk aversion, choosing different types of options to address and evaluate biological endpoints. Normally what we would do is take a look at the information available for a device and all of the biological endpoints required for evaluation, and then either explain why those endpoints are already addressed with the previous testing, or prescribe options to address the gap. So you can see, for example, if you have a device that has permanent contact with tissue bone, then you have this long list of endpoints that are needing evaluation. We'll go one by one and assign a way to address that gap if it is a gap. So for example, for cytotoxicity testing, this is or cytotoxicity as an endpoint for evaluation. This is something that's really a straightforward biological test. It's not really burdensome. It doesn't take a long time or cost a lot of money. If cytotoxicity is a gap, then we basically always are recommending to do this biological test. For sensitization and irritation, this is something that's kind of an interesting point. So these are in vivo in the U.S., at least for the FDA. These are in vivo animal tests. In Europe, though, the European notified bodies have been really open and accepting of using in vitro alternatives for sensitization and irritation. And I think that some companies, because we're so used to the FDA's aversion to the in vitro options for these, have kind of, been feeling like that's really their only option, but really it's not. So sensitization and irritation, this is one of these things that for a European notified body or an MDR remediation, first of all, we can do a written evaluation to address this without testing. Oftentimes, if the materials are very well known and all the contacting materials are known, then these can be screened and explain why there's no sensitizers or irritants there. If there is a gap that can't be addressed, pen and paper, then we would recommend the in vitro tests for those to spare animal life on that point. And these tests we believe are better than the in vivo tests. So sensitization and irritation, there are options there to address those. Material-mediated pyrogenicity, this is one of these that if there is a gap on that point, we have had some success, with different types of regulators defending that testing isn't necessary with a written evaluation, but we often will recommend just doing the biological test for that one. For the systemic tests, like these systemic toxicity, subacute, subchronic, chronic, and so on, right? So now there really are a lot of options in the way that you address this risk, and this comes to another point where we've seen and learned a lot over the last year, right? So depending on the device type, it may be possible to do a written evaluation for these endpoints without doing any type of testing, right? So sometimes you'll have a device type like an anodized titania piece, and this anodized titania piece has excellent evidence associated with it that it's totally clean. And based off of that, we can say, okay, there's no surface residual chemicals on there that could possibly result in repeat and chronic exposures to any chemical that might cause any of these endpoints. So it can be done pen and paper without any testing. More often than not, devices are more complicated than just a single material like anodized titanium. And so we see a lot of folks doing chemistry testing followed by a toxicological risk assessment to address those endpoints. And here's where what we've been seeing over the last year is that there's still, even though we've had the draft ISO 10993-18 out for some time, so we've known what's coming. There isn't a lot of consensus in industry and across to notified bodies on the details of how that chemistry testing should really be done. So, for example, we see some companies doing very, very detailed and in-depth and I would say very expensive chemistry testing in order to address these biological endpoints. So big projects with really a lot of detail, and we see some notified bodies like BSI that have gone really, really deep into the chemistry and are starting to ask for lots and lots of detail. Sometimes, maybe a little bit more detail than what we feel is truly necessary to predict toxicological risk, but still going very deep. While on the other side of the spectrum, there's other companies that are doing very, very little chemistry comparatively, so much lighter, maybe just like a GCMS screen, and some notified bodies that are accepting those types of super light chemistry screens. So our observation as a lab has been that the type of chemistry testing that needs to be done and the level of detail and depth that is needed is highly dependent on which notified body the data is going to. And one of the things that we try to do, so we are ourselves as an advisory services group, we've built relationships up with auditors and reviewers in a bunch of different notified bodies, BSI and TÜV-SÜD especially, and so we have conversations with them about what types of things that they want in their chemistry testing and what do they expect, and we also recommend that sponsors, especially with a newer notified body or one that we have less experience with, that they try to have some of this conversation with their notified body ahead of time. Now, I know that notified bodies don't usually do a pre-sub process, at least not in the sense that the FDA does. Especially since if you do a pre-sub with a notified body, and this is something that you can expect to pay for. But you still can, while a notified body isn't going to be making recommendations on the details of chemistry testing that they expect, they can tell you if they would take what you proposed or not. Or they would say, yes, this is to us, this is in compliance or not in compliance. So this is something that has been really valuable to do ahead of getting too deep in a bunch of a very long and expensive chemistry project. It makes sense to check with the notified body ahead of time to see if they agree with that level of detail is necessary. So as you go through, you identify gaps in the biocompatibility files. With those gaps, then you go and we've been prescribing different types of strategies to address those. What should be clear is that there's always options for each of these. Of course, from my perspective, using a risk-based approach in the spirit of the 10993 series, really any time that you can truly protect patient safety and adequately address risk without testing, to me this is a favorable path to go forward. So I would look for those types of opportunities all along. Another part of this MDR remediation strategy, especially if you're going to be doing some testing, is this family grouping process. So one way to minimize the amount of tests that really need to be done is by making sure or designing sample groups so that those test results can apply to as many different product numbers as possible. And one of my favorite examples are orthopedic sets. So these are things like plates and screws that can be used together. And sometimes you have a situation where you could have 2 or 3 different plates that are used at the same time in a single surgery, and they could be used with any number or different combinations of different screws. And so if you start to iterate over all the different possible combinations of these parts in a clinical use, there's a huge number of clinical uses that could really be possible. And that's one of those cases where it's like, well, do we really need to prove every type of clinical use, or do we need to check each screw type or what? And if you're careful, you can usually design what we would consider to be a worst case clinical use, taking the most number of plates and the most number of screws that would be used clinically. And then if you take a look at the differences between things like different screw types and different plate types, it's often arguable that certain types within these categories are equivalent from the perspective of biocompatibility. So what could be a whole bunch of line items and different clinical use configurations, it can be condensed down often to just 1 or 2 sets of parts that represent the entire collection. So I would say that this has been one of these things as companies have gotten neck deep in the MDR remediation process. I think that they've realized that they can use family groupings to their advantage more and more, and that's really limited the amount of testing that needs to be done. Okay, so let's shift gears just a teeny bit more and talk about another thing that we've learned a lot about over the past year, and it's one of these that at the beginning looked like tons and tons of testing, and then over time, it seems like we've been able to address a lot of this with maybe less testing, and that's with these CMRs, right? So GSPR 10.4 in the MDR specifies that devices or parts thereof or those materials therein, so the materials that make up a medical device, they really should be free of these substances which are carcinogenic, mutagenic, or reproductive toxicants. So that's CMRs. And in addition to that, they should be free of these endocrine disruptors. And so really, there's some code there listed that actually refers directly to chemical lists that you can go online and download it in Excel format. So this one, Part 3 of Annex 6 to Regulation 1272, that's the CLP Annex in the EU. And if you download that, you can download an Excel file and filter, so you just view the CMRs, right? So basically, GSPR 10.4 is saying, okay, you can't have anything that's on that list of CMRs at a weight percentage greater than 0.1% by weight, and the same applies to endocrine disrupting chemicals or the subset of the REACH chemicals that are endocrine disruptors. So this requirement at first has seemed really, really burdensome, and I agree it can start to get a little bit problematic to address this. But on the other hand, once we've gotten into this, like REACH, material suppliers really do have an obligation to disclose and relate whether or not the materials that they're supplying a medical device manufacturer, whether or not they have these chemicals at concentrations greater than 0.1%. And the intention of this, and as far as we've understood from our conversations with notified bodies, is that really the idea here is that the medical device manufacturers should be asking their suppliers for this information up front, and the suppliers are really supposed to be ready and able to give this to their customers. And at first, a lot of the conversation was that yes, we've asked our suppliers, and they say that the formulation is confidential, and they just won't tell us whether or not there's any CMRs above 0.1%. And we heard that a lot, but now over time we've been seeing more and more suppliers willing to make these CMR type of statements or certificates where they simply just claim, no, there aren't any chemicals on this CLP Annex 6 CMR list that are present above 0.1% by weight. And so this is the general process that you see on your screen here that we've been using to address CMRs, or that we've been advising companies to use as they address their CMRs. So the first step in this process is to really make sure that you're asking your suppliers for this type of certification, whether or not there are any CMRs in there. And I think that notified bodies know that this really is the best course of action, and really I view it as a requirement. So asking your suppliers and documenting that you've asked your suppliers for this is something that you really must do. So once you've asked your suppliers if there are any CMRs above 0.1% by weight, then you have a flow chart here on how you can react, right? So if your supplier discloses to you that yes, you do, there are CMRs in those materials, and it's at a concentration greater than 0.1% by weight, then the path is pretty straightforward. You would just label for that and have some sort of written justification for why that chemical is acceptable to be inside the device. If you're in a situation where the supplier won't give you or can't give you CMR information, then the path that we've had to go forward is to do a chemical characterization process where we'll do an extractable study, like an exhaustive extractable study on a device or the device materials So we've done a lot of these on like resin beads, and take a look at what chemicals under exhaustive conditions can really leave the device. And if there's any CMR found that can leave the device at concentrations that would be anywhere close to 0.1% by weight, then we reflex from there and say, okay, there might be a CMR above 0.1% by weight, and additional work might be necessary to really determine if it's over that threshold. But more often than not, what we find is that even when we extract exhaustively, that there aren't any CMRs that we can find that have left device materials. And what we would do at that point is make a statement saying, look, we extracted exhaustively. There weren't any chemicals found that are a CMR, and and therefore we don't expect any to be in the device materials or the device above 0.1% by weight. And this has been well accepted so far. Granted, there's very little experience with these strategies going all the way through the notified body review process, because there's so few devices that have been submitted and accepted under the new MDR. But in our conversations with notified bodies over the past 2 and 3 years, as we've presented this strategy to them, they've been very well accepting of this and indicated that this is in line with their thinkings to address that. So at this point, I think what I'll do is I'll turn the time over to Jennifer. I'm super happy and actually flattered that Jennifer was willing to co-present this presentation with me. So I'll go ahead and give her the time for the remaining of this presentation so you can hear a true industry perspective on this MDR remediation process. So I'll hand it over to you, Jennifer.
All right, great. Thank you so much, Matt. Thank you for the invitation to co-present with you. Again, my name is Jennifer Braunschweig. I'm an engineering director in the neuromodulation business within Medtronic, and I lead the Materials, Biocompatibility, and Applied Chemistry group. So as we have been working through our approach to MDR remediation, Matt hit on a lot of great points. First thing is to review and identify. Review existing testing, documentation, and identify any gaps. So as Matt mentioned, a gap assessment that you may document in a biological evaluation plan to establish what are the gaps that you have in your body of evidence for your device. Next step is execute, gathering information, executing testing to fill those gaps. So if it's reaching out to your suppliers, gathering manufacturing information, we'll talk more about that on the next couple of slides, but that's your execute phase based upon your gap assessment. And then the last piece, document. Tell the story for your product. So pulling all of those pieces together and telling the story for your product to the regulatory audience. So the first step of review and identify, like we stated, assessing and identifying the gaps for your current product. In May of 2020, TÜV provided a checklist. So this is a new tool that you can use to see what are some of the required evidence that, in this case, TÜV, but a notified body would be looking for when it comes to biocompatibility. So looking at that checklist, when we talk about device description or variance, so a variance in neuromodulation could be the length of a lead, that it comes in different lengths. Could be the size of the connector. Device size, so you see there a picture of our implantable targeted drug delivery pump. That pump comes in two sizes, 20 ml and 40 ml. Intended use per the IFU. Again, that understanding for your staff who is assessing biocompatibility about the instructions for use, it seems like a very simple statement, but it's complex about how the device is intended to be used, and then how does that identify your biocompatibility strategy, and how does that influence your biocompatibility strategy? What are the body contacting materials in that device based upon its instructions for use? What are the patient populations? Are we talking about pediatric patients? And that brings in a different sense of the use. Components and raw materials. So again, that is a requirement that we must know what everything is made out of, including additives. So my example on additives is years ago I worked on a catheter project where the catheter was green. After much digging, found out that that green colorant was actually the combination of 3 colorants: a green colorant, a yellow colorant to make it a different shade, and a white. So there you would look at something and say, oh, there's a green colorant in there, but really there's 3 different colorants that made up that shade of green. All of those need to be identified. We need to know the name, the actual chemical name of the colorant or additive in products. Suppliers, identifying who are the suppliers and throughout the supply chain for the products and for all of the components and materials that go into products. Manufacturing locations and process flow, including processing aids. So what are the different manufacturing steps at each of the locations for manufacturing? And what are the processing aids that come into contact? That includes things like tooling and any other aids, tooling, tweezers, anything else that comes into contact with the product. Shelf life and handling. Is there an impact to shelf life on your product? How are those products stored and handled? Matt mentioned qualifications for personnel performing the assessment. What are the qualifications for the people who are performing the assessment? That needs to be part of that. Chemical characterization. Do we have chemical characterization data? What were the samples, conditions, test lab qualifications? Was biological testing performed? And the same thing there. And then also a list of the physical properties: size, shape, roughness. And then finally, like Matt talked about, CMR, endocrine disrupting substances. So these are all the things that we want to look at to assess our gaps in our review and identify phase. Moving forward into the execution and documentation phases, this is where we want to gather that information. So we want to have robust documentation on the manufacturers and documenting the materials, the manufacturing process, the suppliers. Pulled together, we try to pull that together into one source or one location as best as we can, so that we have that information and it is then referenced in our biological evaluation reports. Reviewing the device description and reviewing that with the appropriate folks in the organization, whether that's design, systems, your clinical folks, to really understand that device, how the device is used, therefore, what is our biocompatibility strategy, and what is the identification of our tissue contacting materials and components. If we find that there is a gap large enough that we need to fill it with testing, then initiating protocols and executing testing for chemical characterization, and then a subsequent tox risk assessment after that. Any biological testing, and as Matt mentioned, cytotoxicity is a common one where that's one that we run often to fill gaps. Physical properties, taking those products, looking at surface roughness, shape, size, and thickness. This actually can then be a benefit to your product moving forward. On the right-hand side, I've got a picture of a newly released Medtronic device, the Percept primary cell device for DBS, where we do talk about the size, the shape, the contour, and how that positively impacts the patient based upon the shape and size of the device. Having our gap analysis around hazardous substances. So, as Matt talked about, working with suppliers to gather that data on CMRs and endocrine disruptors. And what we've really found at Medtronic is that we really value our relationship with our suppliers and seek out suppliers who are willing to share and provide information to us. They become valuable partners in our process to not only design products, but then to manufacture and be long-term suppliers to us. And that also stretches to not only neuromodulation, but other businesses within Medtronic. So, the first course of action is working with our suppliers for hazardous substances information. We may go forward and decide to do testing for hazardous substances if we cannot get the information that we need. One caution I would have there is to make sure that you have confidence with the testing labs and the methodology. We did move forward initially to send a bunch of products into testing. We found that the laboratory we were working with, after we had done some testing, we weren't as confident with them in their methodology. We questioned some of the results that were coming back. So again, if you're going to move forward into testing, make sure that you have confidence in your lab, confidence in their methods. Also, if you're going to move forward into testing, make sure that you have somebody who is looking at the test data when it comes back. Again, we started a bolus of testing. Information came back from the contract labs, and then we needed to have the folks to really dig into, analyze, and interpret the information as well. That would be the final bullet point there, the staff to analyze results and address any issues. Are there questions? Are there questions on the reports? Are there questions about the test methods as well? And then the final point is then pulling all of this into a document that tells the story for your product. Remember to include the qualifications for the evaluators and for your laboratory. So laboratory certifications, what quality system you're using for the different testing, if that testing is being done internally. We also have the making sure that the writing in your document is clear as you move forward and written for a regulatory audience. Can we tell the technical story of our product for a regulatory audience? With that, that is the end of my part of the presentation.
Okay, excellent. Well, thank you so much to Matt and Jennifer for sharing your expertise. So now we're moving on to the Q&A portion of our event. And as a reminder to our audience, to participate in the Q&A, just type your question into the text box located to the right of the presentation window, or click the Q&A icon at the bottom of your screen. If we're not able to answer your submitted questions during today's webinar, We'll be sure to share them with our speakers who can reply to you offline. To get to our first question, if a safety assessment of an extractable leachable additive is conducted and PDE is established with respect to one MD, is it required to establish safety of the same extractable additive in other medical devices? And
hi, Daphne. I'll take that one at least at first, unless Jennifer wants to chime in at the end. Putting my toxicologist hat on here. So this is something that we do day in and day out. The safety assessment of extractable compounds found from medical devices, and part of that process is establishing a permitted daily exposure, or a PDE. But if you go through this process for one device, and let's say that you find urasemide from the device, or DEHP, something like that, and then through the safety assessment process, you establish a tolerable exposure, or a PDE, then that gives you, in the context of that one device, it lets you know whether or not it's that amount is acceptable. But it may or may not be applicable to other devices, because other devices can have other clinical uses, right? So I would say in some cases, let's say like a catheter that's an anesthesia catheter. Some anesthesia catheters, only one is ever used at a time, but in others, 4 can be used concurrently. And in some devices, it's really truly limited, like less than 2 hours or so of cumulative use. But in a different device, it could be more of a chronic use type situation. So I would say that it's generally not acceptable to take one PDE and apply it to a different device unless the clinical use situation of that device is really exactly the same.
Okay, great. Thank you. Jennifer, anything to add, or?
Nope, nothing to add. Thank you.
Okay, great. Okay. Matt, you mentioned there was a means to use in vitro methods instead of in vivo for certain tests in Europe. Will that be replicated in the U.S.?
Yeah, so there's in vitro methods for irritation and sensitization that are currently available. We're running the in vitro irritation method right now inside Nielsen Labs, and the in vitro sensitization method is being worked on. So those are acceptable in Europe. We really do hope that the FDA will fully recognize these soon. We know that they're on the path to recognizing these in vitro methods. Though I would say yes, I fully expect that in the future this will be accepted in the U.S. The status right now, though, is that they're not.
Okay, thank you. Another question for you, Matt. Do you have comments on tests favorable for evaluating or justifying why CMR 0.01% or 0.1% may be acceptable, particularly in context of other biocompatibility testing that may meet acceptance criteria?
Yeah, that's a good question. So there are a lot of devices out there, especially PVC devices, that have CMRs above 0.1%. So PVC commonly has a plasticizer in it, diethyl hexylphthalate, DEHP, and it's often at concentrations greater than 0.1% by weight. So in that situation, the GSPR 10.4 simply says that the presence of that compound needs to be justified. Now, to justify it, I wouldn't really point to a specific biological test. Rather, what I would do is point to chemistry and a toxicological risk assessment. So if you do an extractables study on the device that contains a CMR and you detect that CMR at a certain amount, then a toxicologist can claim conclusively whether or not that amount actually could be harmful to a person or not. And there's lots of situations, or I would even say the vast majority of situations are that, while there may be a CMR above 0.1%, if you take a look at this mass percentage doesn't correlate to the human exposure at all, right? If you take a look at the human exposure, actually the dose makes the poison, and the daily exposure to these would be so low that it's not a concern from a safety perspective. So I think the way to justify that is with toxicological risk assessment.
Okay, thank you. Another question. Of the gaps in biocompatibility you identify through MDR gap assessment, how often are those addressable through material review?
Yeah, this is Jennifer. I would say that material review should be your first course of action. I think you can address a number of gaps with reviewing materials, your internal documentation, as well as working with suppliers. And that then moving into testing would be the next level after you complete that material review and find that you still have gaps.
Okay, thanks, Jennifer. Anything to add, Matt?
No, I agree with everything that Jennifer just said. I think that that most of the time when we identify these gaps, material review can be really useful. That's been our experience over the last couple of years. Is actually the information out there is a little bit better than expected, so that it's not that we're just testing all the time.
Okay, thank you. We have heard of lots of different approaches to addressing CMRs, from testing each lot of resin beads or testing devices, or even no testing at all, just relying exclusively on supplier documentation. What are you doing, and what is the most common approach? And I think that's for Jennifer.
Yes, so the most common, I would say the most common approach after some learnings is to review your documentation first, work with your suppliers to gather information, and then mine the information that they have and are willing to provide before going forward into testing. Like I said, we initially had gone forward with testing, and then found that the lab that we were using, we had questions about their methods and the results that were being provided. So my heat of caution is first look at your own material documentation that you have, your supply, and then work with suppliers to gather any documentation that you may not have on hand, and then move into testing if needed.
Okay, great. Okay, excellent. Well, that's all the time we have for questions today. I do want to thank Jennifer and Matt for your time today. We do appreciate you sharing your expertise on this topic. And thank you to our sponsor, Nelson Labs, very much. And as well as to everyone in our audience. We appreciate your attention and your participation. Within the next 24 hours, you will receive a personalized follow-up email with details and a link to today's presentation on demand. Please feel free to invite your colleagues and peers who may not have been available to listen to the event. This webinar is copyright 2020 by Informa. The presentation materials are owned or copyrighted by MDIQMed and Nelson Labs. The individual speakers are solely responsible for their content and their opinions. On behalf of our guests, Jennifer and Matt, I'm Daphne Allen. Thanks for your time and have a great day.